background and training to this subject. Dr. Gale?
Thank you. Thank you for this very warm introduction. It's my great pleasure to be here today. You may already have noticed my French accent, so it's the first time I give a presentation while people are having dinner, so I must start with using my good French. My presentation is entitled Clinical Updates in FLASH Therapy, but as compared to the previous discussion you've had with Professor Meyer, FLASH is a very newborn modality. And of course, we don't have that much data to date to share with you. So I think the goal for me today is to rather share with you our overall vision and strategy that we have developed over those last 5-6 years to understand how we expect to bring FLASH to the clinic, where we are at, and what remains to be done. This is just a disclaimer as to remember that FLASH therapy is still under development and not available for commercial sale. Okay, so as you know, and for physics reasons, the rationale for a sparing advantage with proton therapy is clear and relatively well accepted. And it's also true that conventional radiotherapy has made tremendous technological progresses over the last decades. And for this reason, the emergence of proton evidence has competed with photon-based evidence. Still, we now have a well-identified scope of indication for which proton therapy brings a benefit. And in addition, as the proton community has substantially grown, so did its ability to launch clinical trials. And we expect additional indications to be validated within a short-term horizon. But I think that the message here that I want to first discuss is that the proton community has organized itself quite well over the last 5-10 years, which enables us conducting new type of trials, but also new type of research. And it's quite recent that in the field of proton therapy, we are actually able to study radiobiological rules at the level we did for FLASH therapy. As a consequence, there's been an increase in the number of clinical trials for proton therapy in the past 3-5 years. And there have been a significant number of trials, especially in brain, CNS, breast, liver, lung, and head and neck. For these location, the challenge at the technological level are slightly different. For instance, for brain, spinal, CNS, we need high precision, and this has been especially enabled by proton pencil beam, as well as TPS optimization. While on the other hand, we have also developed tools over the last decades as to deal with the motion management, including fast scanning solution, respiratory management, or continuous imaging fluoro-like solutions. And here the message is that for proton, just like it's been the case for photon optimization over the last few years, most of these disruption have been achieved by pursuing a technological development effort. So this is really the DNA of radiation oncologists or radiation biologists. Most of the optimization has been driven by technology. For FLASH, it's a different story because FLASH is not defined by the technologies. It's a biological effect that is serviced by the technology. So it's a different approach. And I'm very glad that Varian, for the first time in its history, has actually started investigating biology with the same importance it was developing technological solutions. The FLASH effect is defined by two outcomes that must come together. The first is the observation that for a given dose, when we increase the dose rate of delivery, the expected toxicity is drastically reduced. So this is one of the first papers that I've reported on big animals, this observation. You see on the left side the skin of a pig that had been irradiated with conventional electron radiotherapy at 40 to 38 gray. And after a few months, we observed the expected necrosis here on those dark spots. The same dose were applied on the same pig on the contralateral side, but the expected toxicity were never recorded despite more than 1.5 years of follow-up. The second part of the definition is that even if you effectively reduce the toxicity burden, you should maintain, obviously, your ability to control the tumors. And this was also early on reported from a cat study. It was a dose escalation study, I think, scaling up from 30 to 44 gray as a single dose, while the clinical standard was 10 fraction of 4 gray given twice daily. Out of this first dose escalation study, authors have not reported any toxicity despite a 2 years follow-up. There's been a phase 2 clinical study after selection of a single dose of 32 gray. And this second study performed by the same teams this time reported much more toxicity. And interestingly, the only difference between those two studies was the beam orientation. So in the first case, the beam orientation was quite parallel to the animal, while for the second study, the beam orientation was perpendicular to the animal. And while the authors have conducted retrospective dosimetry studies, because this first study was not performed with treatment planning before, they observed an overdose with hotspots of about 30% due to the nature of the electrons. And of course, this has raised the message that FLASH is super interesting as to spare the toxicity, but FLASH is not a magic bullet. FLASH also induced toxicity. We have some upper limit thresholds, and we need to define them before we can design safe clinical trials. So as I said, the technology serves FLASH, but what do we talk about when we discuss increased dose rates? So as to illustrate how faster we must go, I often use this cartoon that shows you typical speed for a walking man to a satellite turning around the Earth. If this has to be your conventional dose rate, which is typically delivered from 0.03 to 1 gray per second, the FLASH dose rate that has been theoretically determined between 40 to 100 gray per second would be here. So you can understand that technology-wise, it's a huge gap, but in terms of safety, it also comes with new constraints that we had to deal with. Overall, the promise of FLASH is always the same within the therapeutic window, meaning pushing to the right side this toxicity curve that we have to deal with. I want to stress out that this threshold of 40 to 120 gray per second has not been empirically demonstrated. We have upcoming data showing that for some specific organs, it might actually be much lower, which from a feasibility standpoint is quite interesting. And those data will be presented in a couple of months at ESTRO. In practice, this widening of the therapeutic window can be established on preclinical or translational studies. And we are basically repeating the type of studies that were very popular 40, 50 years ago, especially performing dose rate escalation, etc. So this is a very old format of study where you can observe skin toxicity on rodents. So again, these are small animals. And we report the overall damage at early time after radiation, which is typically 2 to 4 weeks after radiation, up to 6 months after radiation. In between, we also record the tumor control ability. And what you can see is that in both cases, in terms of toxicity, when we apply ultra high dose rate, we observe a clear sparing effect that is both true in acute and late setting, but with different magnitudes. It's also interesting to see that the overall sparing factor, as we name it, is a function of the dose and has somehow an upper or lower limit. More of these studies are currently being conducted, and while the first FLASH paper was published in 2014, we now already have more than 2,000 peer-reviewed articles that cover the technological aspects, the dosimetry aspects, as well as the biological aspect. And as to align with what has been said during the previous presentation, it is true that to ramp up and scale up evidence generation, the community must be extremely organized. So I think, and it's actually a key difference with the field of SFRT, we are a much younger field, but on the other hand, we are also organized from the beginning. We don't have to re-standardize the practice because the evidence is so new. And finally, out of these massive collective effort across the world, we were able to include the first patient from a clinical trial in 2020. So it's very fresh, but on the other hand, it's also quite impressive that we achieved first step to clinical trial in such a short time. So how do we do this? So currently we have created a Flash Forward consortium, which gathers multiple institutions, about 30 institutions, more than 200 members. Not all of these institutions are necessarily Flash enabled, so that they may not all be able to conduct biological studies, but one way or another, they are all able to bring their stone to the wall. As of today, and again, this is aligned with what has been said in the previous presentation, we've been able to reproduce the flash effect across centers using different technological devices, across organs and across species. So we have data for mouse, of course, but also rats, fish, mini pigs, and we start now having data for human. There are some top organs for the flash effect, especially the skin, the lung, the brain, the gut. But it doesn't mean that the other organs are not suitable for flash. It's just that on those small animal systems, it's more difficult to evaluate the toxicity of the bone, for instance, or the esophagus or the kidneys. So we have a lot of proof of principle and proof of concepts data, but there are still some gaps for dedicated organs, which has some impact when we design the clinical trials for the future. So this slide is quite heavy, but it kind of pictures precisely what has been done, where we are, so that you understand what we can do at the clinical trial level and what still needs to be supported by additional evidence. I would say that from a biology standpoint, 80% of the papers that were published are proof of concept papers that usually investigate a single dose, which is often irrelevant from a clinical standpoint because those animals are more resistant than humans. Single organs, single parameter study, and healthy tissue sparing and tumor efficacy are always reported. So this is good as to create an enthusiasm, as to create structure around a given organ, but this is not sufficient to help us design clinical trials. To go further, we had to increment some complexity on those studies, especially finding LD50 for several organs. And we now have those data for the skin, the muscles, the subcutaneous tissue, the lung, and we expect to have spinal cord injury from a mini pig study by the end of the year. But we also need to define what are the thresholds for those rates. And again, I think that by the end of the year, we will have a clearer idea of what is a prerequisite for FLASH. We have multiple organ investigation. Now, we also, so one dose rate does not fit them all. So it means it's not just about going faster. The way we deliver the beam, whether it's coming from electron or proton, it's very different. So we have to understand the nature of the beam. What are the key parameters to elicit the flash effect? Should we deliver it continuously with a given number of pulses, etc.? So as to conduct what we call parametrization studies, and there are multiple by now, we had to downscale our experiments, and we've been working typically, for instance, on ex vivo tissue, where we were collecting tissue, growing them on culture. And I know that some data were presented last FRPT meeting in December, where, for instance, we've been able to demonstrate using surrogate biomarkers a flash effect on human-derived lung tissue. So basically, they took some biopsy from the lungs, submitted it to flash versus conventional dose rates, and they have observed on a given dose range and a given dose rate range some sparing effects. So these were the very first data reporting a flash effect on human tissue. And this is where we are now. So constraints, identification, and mitigation strategy. Basically, there are a few things that came out from this literature, and we have some bottlenecks to validate our checklist before we can go further in the complexity of the trials. For instance, a pending question is whether there is a minimal dose for a flash effect. We don't know if the flash magnitude for a single dose of 15 gray would be equivalent to a single dose of 10 or 2 gray. And of course, if you think about flash in the frame of a typically fractionated schedule, this question is of prior importance. Again, some data are currently under submission and under review, showing that using a typical fractionation of 3 gray on rodents and applying 10 fraction, which is already quite impressive because it's difficult to anesthetize those animals, the sparing effect was conserved. So this will set the lower limit of observation for flash to 3 gray per fraction. We are very close from the 2 gray we want to achieve. What about beam interruption? All these studies were performed with a single beam. We need to know if once we will interrupt the beam so that we can have several entry beams per patient, whether the magnitude will be the same. What about the classical biological rules? We don't know if the way, for instance, we calculate the bed or the way we convert a single fractionated scheme to hypo or hyperfractionated schemes is equivalent. We need to manage the entry dose, and we need to learn more about fractionation. So these are currently the major studies being performed, and we do hope that within 1 to 3 years we'll have more data to sustain clinical trial design. I will skip this part because it's more about biology, but this leads us to clinical trials. At this stage, there are things we can do, and there are things we need to wait for. The FASTO1 has been the first variant clinical trial using proton, and the scope of this trial was to include 10 patients with metastasis on the extremities and treat them with a palliative intention. So it's not a design where we do expect any kind of toxicity. So the purpose was really to determine, are we able to date with the current technology to deliver a dose rate, record the dose rate, and make a treatment planning solution in a safe manner and efficient manner. All these patients had to be treated in less than an hour. So all the primary endpoints of the FASTO1 have been validated with success, and we've not reported unexpected effects due to the flash treatment. The second clinical trial, which is not a variant trial, is currently being held in Lausanne, Switzerland, and it's an electron escalation study where they are testing two things. The first thing is the dose escalation, I think from 22 to 34 gray. And the second thing is the volume effect. So there are two arms, one arm with less than 1.5 cubic centimeter and one arm with bigger tumors. So I think this study will validate within a short time frame what if the magnitude of the flash effect that we have reported so far on animals can be translated in human. So this is a very important study that is well advanced in the accrual of the patients. And finally, we have FASTO2 that is ready to kick off, where we will also include about 10 patients, but this time we will treat the patient with metastatic occurrence in the thoracic cavity. So it's also a feasibility study, but this time we go a little bit further in the complexity, especially from a treatment planning standpoint. We are not only interested in proton, we are also interested in electron or other modalities enabling flash, but maybe it's good to explain why we started with proton. That is, the dose rate definition is very different from one modality to another, and this is what is captured by this cartoon here. If you consider dose delivery from a proton standpoint, each time you deliver one spot using the PBS technology at the voxel level, you deliver the entire dose. However, it takes a little bit more time to deliver the treatment over the whole field as you need to move the spots. Meaning we have two dose rates very relevant for proton. The first dose rate is what we call the PBS dose rate, which is at the voxel level at how fast we deliver the dose, and the field dose rate, which is how much time it takes to deliver the whole treatment on the whole field. And of course, the trick here is that the PBS dose rate is completely independent from your volume, so from the volume of the target. If the volume is big or small, the PBS dose rate will always be very good. While if the volume is very high, then the field dose rate will be reduced because we need some time to cover the whole area. And by putting the effort on proton, we took the chance to figure out which of these two definitions is the most important, because for electron-based flash radiotherapy, we don't have the voxel level. The voxel level and the field dose rate are actually the same because the whole area is being treated in the meantime. So again, we have launched several studies as to determine which of these parameters is the most important, and it appears it might be the PBS dose rate. So those data have not been published yet, but they will be presented at ESTRO. A lot of things will be presented at ESTRO this year in Vienna in a couple of months. And so based on these, we've made the choice to follow the step as it should be done when it comes to implementing a new technology. For flash, we want to go as fast as possible, but as slow as necessary. So our top priority is, and has always been, the safety of the patients. That is why the first trial has not been testing a dose escalation. In the end, we do expect to test several indications. We have pre-identified a list of priorities, but the priorities is also governed by the pre-existing preclinical and translational evidence. So it basically fits with what has been done at the research level. We are quite proud as a device company to have entered the biology research field for the first time, and I think it's quite unique for a radiation device company. And this has been granted by a few successes over the last few years, especially the FAST-01 series. The enrollment completed in a very decent time short frame. We've managed to keep all the dose rate over 56 gray per second, which is way over the current threshold limits as defined by the literature. We've already been able to treat some relatively big field size, and we have not reported any serious adverse events to date. But what is even more important and interesting is that the overall community has very well understood our strategy and now also understand what are the key milestones we must achieve for the next few years. And that's why we know that in the coming future, as people will gather and organize themselves, we have the chance to early on standardize the whole practice so that we go in one direction from start. And so this is the result of FAST-01 that has been published in JAMA Oncology a few months ago now. And it has triggered a lot of reactions, and we are very glad of it. This is the timeline we've had over the last few years. So you see that the Flash Forward Consortium is only 5 years old, let's say, and a lot have been achieved over those last 5 years. We've been deeply involved with FDA regulator and regulation in general as to make and obtain an IDE approval and make one of our centers able to enter the clinical trial room. So to date, only Cincinnati is able to do this, but we hope that more and more centers will enter this process, for which we bring support. We started the enrollment and concluded in 2020, and we expect to trigger FAST-02 and enroll the first patient this year. I will skip this. So as I said, as we move forward, we will need more and more centers to be enabled and more centers to be involved, both on translational, preclinical, and clinical research. There are multiple steps to go through for this, but in all cases, we need solutions. So we need to be able to guarantee the quality assurance for FLASH. We need to be able to perform treatment planning. We need support whenever the technology needs maintenance or whatever, etc., etc. And so Varian has designed a complete set of solutions that is available as to help and support any centers interested in FLASH research. I will have a very quick word on this because these are future upcoming research for FLASH that kind of shows some synergies with the previous discussion. So you've just been introduced to SFRT, and one of the questions that has been asked just before is, why don't we go for small tumors? So I love the answers that were given, but I think there is another aspect, which is the cultural aspect. It's difficult to engage and enroll physicians to treat a lesion without covering up the whole lesion. If for a small tumor, you kind of know you could have treated it and have some decent chance to achieve tumor cure. For SFRT, you have different levels of SFRT. And for proton beam, we know that we can reduce in a size range the beam and the valid dose to a minimum of less than 1 millimeter. And the one advantage of proton mini beam would be that we could challenge this cultural issue by beneficiating from SFRT on surrounding sparing tissue, while using the nature of the proton beam to cover the whole GTV or CTV in an uniformous manner. And so another aspect of SFRT is that the smaller the peak-to-peak distance, and the more likely it is that motion may reduce your sparing effect induced by SFRT. And this has been touched during the previous presentation. How do we manage motion during SFRT, whether from one session to another or even within the same session? And for this reason, we believe that combining FLASH and SFRT offers key synergies, in the sense that we would be able to deliver SFRT in less than 1 second for a lot of indication and a lot of field size. In this regard, and we are already working with some key opinion leaders in the field as to integrate treatment planning solutions for FLASH and SFRT together. And the spectrum we want to cover here, it's what is presented here is a collimation system that enables SFRT at the submillimetric range, and we are already getting prepared as to offer the follow-up solution together with SFRT. I will skip this slide because I think I've already covered everything I wanted to say. So I'm happy to take any questions, but I will just now that you are having your dessert, wishing you a good late evening afterwards.
Thank you very much, Dr. Gale.